everybody. Let me tell you about my favorite topic, longevity. I feel a little out of place. Usually I have a clicker or a microphone to do something in my hands, but yeah, I'll try.
Not so many carpets in the room.
Yes, this is definitely a first for me to present in a very interesting setting, like a boudoir. Want to tell you more about longevity, reprogramming longevity, and this new approach to rejuvenation called partial reprogramming. So next slide. And I guess before we start, just want to talk about the problem that we're solving. And I think I might be preaching to the choir because this is like the longevity track. But this was supposed to be geared to more of a general audience. I was hoping to have people from other tracks maybe joining in and listening, so that we convince them that aging is indeed a problem if they haven't yet realized that. So the solution, next slide. The potential solution to the problem of aging is partial reprogramming, because reprogramming was shown to rejuvenate cells and ameliorate all of the cellular aging hallmarks. And so, but on the cellular level, of course, as reprogramming progresses, cells change their identity to the stem cell state, and also in the process, they seem to be rejuvenated. And the goal of partial reprogramming, or reprogramming longevity, for our purposes, is to harness that rejuvenating aspect of reprogramming and package it up into a therapy that we can use. We are a collection of main cells, and the tricky part is doing this in vivo, in the context of an already formed adult organism trying to bring cells to a more younger state and hope that that results in a systemic organismal rejuvenation. And this slide just shows that all of the cellular hallmarks that were observed to be rejuvenated by the reprogramming process. So I hope you can see this. Hoping bigger screen. But anyways, next slide, please. And just wanted to also point out that this is a booming field. As I'm sure people in longevity know partial reprogramming. They've heard about Altos Labs with their $3 billion, probably heard about Retro and New Limit with their $100+ million funding. But people outside of longevity really haven't heard about this field. And it has a lot of potential to not only create a longevity therapy, longevity therapies, but in the process to create just therapies for existing age-related diseases, because this is a novel approach that can also work with diseases like Alzheimer's, heart disease, etc. Like if we, before having a comprehensive longevity therapy that works for all of our tissues, or most of our tissues, we can pinpoint some disease areas associated with a particular tissue in the brain that hopefully can help us either slow down the onset of existing disease, dementias, heart disease, etc., or maybe even roll back the symptoms of those diseases. Cure is a very dirty word in drug development, but hopefully these therapies will provide a meaningful therapeutic benefit, and if they do, they're bound to be blockbusters, $1 billion revenue drugs, and collectively the market itself, partial reprogramming market, the cellular reprogramming market, could be a $1 trillion market if it works out. If we figure out a way to translate what we see on the cellular level into therapies that can work on an organismal level. Next slide, please. And this is just a shameless plug. I was the first drug developer to recognize the potential of partial reprogramming, and I founded the first partial reprogramming company back in 2017. Okay, next slide. But before we dive into the solution, let's talk a little bit more about the problem of why aging is a problem and what theories of aging there is, why the problem happens. So next slide. Unfortunately, there's no consensus in the field. I'm sure you know, I mean, a lot of you have been on the San Francisco Foresight meeting where Vadim Vladic raised this point that we can't even agree on what aging is, but there are some obvious things that we all recognize about aging. Even people outside of the aging field, we are very good at telling apart a young person from an old person. So we know it when we see it, right? And we can argue about the academic definition of aging, but on day-to-day understanding, we are very good at identifying what aging is. And the biggest problem posed by aging is that it kills us. So ultimately, I think it's a problem. As we discussed yesterday, I think most of us here share this vision that dying and aging is not something that we really enjoy. So we feel this problem is really something we should fix. And the speed at which aging kills us is really, the problem is that it's exponential. The mortality risk because of aging increases exponentially, and it starts pretty low just when about age 10. The chances of dying is about 1 in 10,000 for a 10-year-old person annual. So within a year, your chances of dying is about 1 in 10,000. Then by the age you're 20, 25, it's 1 in 1,000, still very good. But when you're 60, it's already 1 in 100. And when you're 80, it's 1 in 10. So this increases exponentially. Yeah.
It's just statistic on probability to die. Because then when someone is 15, it's not because of aging, but because of some accidents that may happen.
Well,
from natural causes.
The natural causes. So your underlying health status that predisposes you to die on a cohort population-based level at that particular age. So, okay, let's just go to the next slide. And also, besides killing us, aging has all sorts of nice ways to torture us as we get older. There's a whole host of diseases associated with aging that are really unpleasant, and they're all exponentially increase with age. Next slide. And main causes of death, I'm sure you know, it's cancer and heart disease, by far the biggest two categories of the ways humans die. And then there's a bunch of other also unpleasant things like dementias and infectious diseases and so forth. So, next slide. And also another unpleasant thing about aging is that it happens so quickly. We barely get to enjoy our fully formed bodies as we get older, and they start falling apart and pretty quickly. So it's a very unfortunate thing. Like most of our lifespan, we actually spend not in such good health, unfortunately. You could say it's a good thing because we don't have to die from predators eating us, but still, again, from our standpoint in the modern society, it's not very enjoyable to be spending 40, 50, 60 years in a state of not such a good health. Next slide. And the biggest question then is, why does this happen, right? So aging is a problem, why does the problem happen? And again, next slide, there isn't, again, if there's no even consensus on what aging is, there's definitely no consensus of why this happens in the field. There's different schools of thought, and I think you can even plot a sort of continuum of the two different hypotheses of aging with the two extremes. One says aging is programmed, and the other extreme says aging is completely random. And you could also have some middle ground that it seems programmed, but the program might not be purposeful. It might be a kind of shadow of the development program that Michael Bogoslawny calls it. Also, another recent understanding of aging that has come to light is that aging has a very strong epigenetic component. So there seems to be epigenetic, well, you could say, driving force of aging. And whether it's programmed or not, I think it's less relevant for us because for our purposes, since we know now how to modulate epigenetics through reprogramming, the question is, can we then modulate our epigenetics in such a way to produce rejuvenation, epigenetic rejuvenation? So, and again, you don't have to subscribe that it's a purposeful program as long as you kind of agree with the observed facts that epigenetics plays an important role. Then the next question becomes, can we actually do something about it? Can we modulate epigenetics in a way that produces something that we want, which is rejuvenation? Next slide. And just before we go deeper into epigenetics and epigenetic rejuvenation, a brief overview of what epigenetics is. And again, I'm sure most of you know, but for some people who don't, epigenetics is just a broad term about gene expression. There's different genes in our cells, and we have different cell types, but the DNA is exactly the same in each cell type. So this necessitates some sort of mechanism that will make sure that in a brain cell, different sets of genes are active than from a skin cell. And all sorts of epigenetic mechanisms exist. And also, but in the context of aging, we're mostly interested recently in this methylation clocks that have been discovered and seem to be ticking with age in all people in a very similar fashion. And also, during aging, it seems that it's not just that genes are either on or off, there's a sort of like a volume setting to gene expression. And with aging, it seems that gene expression of so-called good genes is dialed down with age, and bad genes is dialed up. And so the question is, can we then bring back the pattern of gene expression in an old person to a younger pattern that is observed in younger people, and expect that this will produce a therapeutic benefit, essentially. Next slide. And we already observed in nature that epigenetics can control lifespan and control it wildly. In social animals, we see social insects, we see evidence of this, that identical DNA can produce lifespans differing by up to 100 times. The queen bee lives for much, much longer than a worker bee, although they have identical DNA. They start out with identical DNA. And other examples in nature, even in non-social insects like monarch butterflies, they don't have social roles, but depending on when the butterfly is born, epigenetics determines if the lifespan will be 1 month or 9 months, depending if you're born in the summer season or in fall. And also similar is epigenetic control of lifespan is observed even in mammals. There's a species of rodents called mountain vole, where similarly to monarch butterflies, if the vole is born in the spring, it lives a short lifespan, but if it's born in the fall, its puberty is delayed, and it starts actually after winter, so its lifespan is significantly increased. Okay, next slide. So the question is then, okay, definitely epigenetics can modulate lifespan in insects. Can epigenetics modulate lifespan in humans? And we have definitely, as I mentioned, have some observations that epigenetics plays a key role in aging, as with the observation of epigenetic clocks. And so the next interesting question is, can we roll back epigenetics and expect rejuvenation as a result? If we actually have this younger pattern of gene expression in older people, will that produce physiological rejuvenation on a cellular level and organismal level? And of course, the hint is that, as I mentioned already, on a cellular level, we do observe physiological rejuvenation. So the next important question, can we observe it on an organismal level? Okay, next slide. And this is where epigenetic rejuvenation comes in by cellular reprogramming. Next slide. And this paradigm really owes itself to two key discoveries. The first, of course, is the discovery by Yamanaka of cellular reprogramming, who have shown that you can actually have this process of full reprogramming of the cell all the way back into stem cell state, embryonic-like state. And the next discovery belongs to the group from Seoul. It was at the time led by Ocampo, although the group was led by Del Monte, but the paper was led by Ocampo, who showed that you can actually use this process of partial reprogramming in the context of adult organisms. Next slide. And what makes it possible, this whole reprogramming and epigenetic rejuvenation by reprogramming, what makes it possible is that reprogramming itself is a gradual process, and the rejuvenation observed in reprogramming is also gradual. So it's not just a binary thing where you jump from one state into the next. Thankfully, this is a gradual process in which you see decrease in epigenetic age gradually during reprogramming, and you start seeing it before the cell is irreversibly transformed. So this means, next slide, that we can find a therapeutic window in which we can observe or obtain rejuvenation before the cell has yet lost its identity. And in the context of a full organism, a multicellular organism like ourselves, it's very important that the cells remain the same cell types that they begin the reprogramming process with, so that our organs don't lose function and continue doing their job. Next slide. And so the paradigm of epigenetic rejuvenation that we are thinking of, and I think most partial reprogramming companies are thinking of, is periodic induction of these reprogramming genes or Yamanaka genes or some new sets of reprogramming factors that we might find in the future that will produce periodic rollback of the epigenetic pattern to a younger state that, if we do this in a repeated fashion, can solve the problem of this exponential increase in mortality risk. So if we're able to periodically roll back our pattern of gene expression to a younger state, hopefully this will also mean that our biological age will be rolled back to a younger state. And if we do it often enough, we essentially hope to freeze our biological age at the same point at which we start the therapy. So basically the goal is, if you take a 30-year-old, you start doing these periodic epigenetic rejuvenation therapies, and by the time the person turns 60, their biological age still remains at 30. So it's not like we magically expect the reprogramming process to turn a 60-year-old back into a 30-year-old, but at the very least, we hope to have the reprogramming process slow down the exponential increase in mortality risk. And I mean, the human increase in mortality is just huge. Like this is a logarithmic graph. So the process is exponential. If you look at it on a linear scale, it just explodes with age. And so this is the biggest problem. And actually the risk, as I mentioned, the mortality risk of a 60-year-old, it's not that huge. For a 60-year-old person, annualized risk of dying is just 1 in 100. And so if we're able to freeze it somehow, that 60-year-old person can expect to live 100 more years. So, not immortality yet, but still from the standpoint of today's lifespan, it's a very good result. Okay, next slide. And, okay, this is the current goal of reprogramming companies, and this is just a brief excursus into how the reprogramming approach formed, how it all began. And before Yamanaka showed this to be false, the dogma was that cell state differentiation is an irreversible process. So people thought that cells start at a stem cell state and kind of roll down this landscape of differentiation. And it was formulated by Conrad Waddington back in the 40s, so this landscape became known as Waddington landscape. Next slide. But this was shown already to be probably false just 20 years later by Sir John Gurdon, who showed that we can actually take DNA from an adult cell, skin cell, and use that DNA, put it in an egg cell, and create a whole new frog. So which means that everything that's necessary to form all cell types in the body is retained in the skin cell. So potentially, this could be a reversible process. Next slide. And this was also repeated essentially in '96 with the Dolly the sheep, the famous cloning experiment that pretty much did the same thing. Okay, next slide. I think I should have 20. Allison, we agreed that. Just present for 20 minutes. And,
okay. We're 5 minutes until the end of the hour.
Oh, wow. Okay. Anyways, yeah, this is the Dolly the ship. Next slide. There were actually several clones after her. Next slide. And the main breakthrough came from Yamanaka in 2006, who showed that he actually showed how to do this, showed how to bring cells all the way back into stem cell state. The reprogramming process was born because of this experiment. Next slide. So the new landscape that had to be updated after this essentially shows that cells can move up and down their differentiation landscape. Next slide. And this then people started asking questions, okay, can we then have epigenetic rejuvenation as a result of this? And the first mention of this was in 2010 by Prem Singh. This is the article in PubMed that first used the phrase epigenetic rejuvenation. Next slide. And the observation that the reprogramming process also rejuvenates cells was first seen in 2011 by Jean-Marc Lemaitre from INSERM, and they have reported this result back in 2011. Next slide. And other groups as well have observed, and this is just a summary slide of which hallmarks of cellular aging are rejuvenated during the reprogramming process. And you've already seen this slide in the beginning. Next slide. And then, of course, the next question is, can we do this in vivo? Can we do this in the context of a formed adult animal? The first attempt at this was in 2013 from Manuel Serrano's group in Spain, and they didn't really succeed. Their mice started dying just 2 weeks after dosing the Yamanaka factors, and that's probably why the title of their paper is pretty pessimistic. It says, reprogramming in vivo produces teratomas, and of course teratomas are these cancerous tumors that we really want to avoid. Next slide. The breakthrough came in 2016 with a paper by Ocampo et al. From the Salk Institute, the group led by Juan Carlos Espiso Belmonte, and they figured out a way how to do this in vivo safely. And main result is they prolong lifespan of these animals, progenitor mice, but still it's a very important result, by up to 50%, depending on which control group you calculate against. Next slide. And they showed that these mice, they looked younger, next slide, and more importantly, they were younger according to several different biomarkers of aging: senescent cells, double strand breaks, inflammation, etc. Next slide. And these next slides just try to dive in. I'm not sure why I have so few time left, so little time left. Anyways,
You started late.
I'd probably. Okay, so yeah, fewer DNA breaks. Next slide. Their blood vessels looked younger, so over time, better histology. Next slide. Other tissues, skin, spleen, kidney, stomach, just the histology of those tissues between the control group and the treatment group. The treatment group looked much better. Next slide. Hair follicles. We know that hair loss and hair thinning, hallmarks of aging. The mice treated with partial reprogramming, they had more hair. Next slide. So what was the key insight of the Ocampo et al. Paper? Next slide. Partial reprogramming. The insight was that you have to do partial reprogramming. You can't allow cells to reprogram for too long because this leads to teratomas. They start forming from cells that have gone too far up the Waddington landscape, or actually just lost their cell identity and have become pluripotent, from which then these teratomas, which is just a collection of different tissue types, form. So in order to avoid that, you need to limit the reprogramming process to just a few days. And next slide. Actually, in the literature before that, there were observations that exactly this happens, that if you let reprogramming proceed for too long, you have teratomas forming as a result. Next slide. But if you limit the expression of the Yamanaka factors temporally, you don't. So if you only have short periods of reprogramming, you don't have teratomas. The cells don't get enough of a reprogramming push to lose their identity. So next slide. And so that was the key issue, the key insight of the Ocampo et al. Group. And the new insight, also recent insight, is that you also, even with that approach of temporally restricting reprogramming to just a few days, there are some tissues you still need to avoid if you want to expand the safety of this approach, expand the therapeutic window. And the recent result, and this is a preprint from the Ocampo group in Lausanne, was that you have to avoid liver and the intestine. And this is the graph that is really illustrative because it shows that if you don't avoid those two tissues, just after 4 days of reprogramming, you have mice start dying. But if you do avoid them, you can expand this process by up to 10 days and all of the mice survive. So just by avoiding those two tissues, you expand the window by 2.5 times. And even after a month of induction, half of the mice still survive. So you understand that those two tissues are really the key ones that you need to avoid. And really, partial reprogramming needs to be tissue specific. Different organs and different tissues have different needs in which you can have rejuvenation as a result of reprogramming. Next slide. Oh yes, and after the Ocampo paper, this really opened the floodgates of research into this approach, and many different papers have come out with essentially validating the observations by Ocampo results. And I just have a few different studies summarized here. For the interest of time, I won't go into details, so let's just walk through them. So that was the previous one was just actually from the Serrano group showing that reprogramming improves memory. Even this is a different group showing that even a single bout of reprogramming can extend lifespan. Next slide. Partial reprogramming can rejuvenate the transcriptome of different tissues. Next slide. It can help with improving heart regeneration in a heart attack model in mice. Next slide. Stroke also can be improved. Next slide. Recovery after brain injury by OSKM. This is from the Sinclair group. You can regenerate neuronal loss and restore vision in mice. Next slide. Muscle regeneration also. So you can see, like, from a therapeutic landscape, there's so many different tissues and potential diseases that can be helped by this approach. That's why I'm so optimistic that this could be a great field if we figure out how to translate this approach. Next slide. yeah, this is just another group showing that different tissues can be rejuvenated by this approach. Next slide. Wound healing. Next slide. You can slow down spinal degeneration by partial reprogramming. Next slide. And yeah, this is just the final slide. And I really like this quote by Juan Carlos Belmonte. This is he was the group leader in 2016 in Salk for the Ocampo et al. Paper, who says that our study shows that aging may not have to proceed in one single direction. It has plasticity, and with careful modulation, aging might be reversed. So I concur. Next, if you have any questions.
Any questions? Great.
I have a question. So both Marinus Sagano and Ocampo and many other persons, right? They did it in project mice, right?
Yes.
Anyone ever done it in
Yeah. Not the lifespan study. Lifespan seems to be tricky, but many other experiments, papers that I showed, they were done in normal mice. Yes. Like wound healing, heart regeneration. Sorry?
But no lifespan.
No lifespan. Yeah, lifespan seems to be tricky, and there was a recent study out of the Belmonte group where they tried doing it in normal mice, and they didn't report a success. So people assume that they probably tried to fail or something. But yes, extending lifespan of normal mice still remains elusive. And maybe one of the reasons because people use transgenic animals where they have all of their tissues have this cassette, but as I showed the recent Ocampo result, some tissues need to be avoided. And if you reprogram just a little too much, mice just start dying. So in the context of a long-lived mice, this could be the problem that really hinders figuring out how to do this safely in long-lived mice. Yeah.
Can you tell us a bit more about why you have to avoid the liver and the intestine? What's special about those tissues?
Yeah, I think it's these are very fast dividing cells. There's high turnover of cells, so it could be that those cell types are very amenable to reprogramming. You give them just a little push, and they become pluripotent. They lose cell identity, and they could form teratomas, or just the organ function can stop functioning.
All right. One more.
Yeah.
It's just two more.
Two more. Okay. All right.
Plus.
Do we have an evidence of partial reprogramming in humans? Is there any trial going on?
Not yet. We have evidence in terms of ex vivo, like some skin cells, some fibroblasts from humans were, in a petri dish. They're rejuvenated and the hallmarks were improved. Mitochondrial function was improved. They took skin cells of a 100-year-old person, several 100-year-old people, and they restored the mitochondrial function in them to the level of young people. So, but in the context of a live human, we're not there yet. We have to do animal studies first.
So there was a paper that just came out a couple days ago about the mycobacterium that causes leprosy reprogramming the liver and causing the liver to grow massively without causing cancer. And it was covered by the BBC, it was in several journals. It's pretty amazing. So, people asked, is anybody capitalizing on this, figuring out how that microbe actually does that reprogramming? Maybe that could be a research direction. And another is ex vivo reprogramming, right? I mean, all of this pharmacokinetic stuff is a challenge, but if you can rejuvenate, say, hematopoietic stem cells ex vivo, that would be pretty exciting. So we should talk about that sometime.
Yeah, sure. And yes, some groups are actually also thinking about the ex vivo approach, and in the context of hematopoietic stem cells. And different viruses and the bacteria, they seem to have figured out some ways of modulating gene expression, and this could lead to reprogramming. And, it's definitely very interesting. Cells have this potential to reprogram. And wound healing is essentially, de-differentiation is the first step in wound healing. And things like limb regrowth in axolotl or other organisms, they essentially reprogram, whatever at the level of the cut, they reprogram cells and then they start proliferating. Sorry.
Okay, no problem. We started a bit late, so it's okay to go over a few minutes, I think, and then we'll just kind of cascade a little bit and give everyone else extra time too. So thank you for Yuri.